Okay, so how are these populations measured? Well, first, some kind of mark is made on the organism. You can see here, like just maybe with a Sharpie, like here's a little mouse with an X on it. Here's some bird bands. These are they're installing like a tag onto the flipper of a turtle or using just like fingernail polish to mark on a beetle. So you mark some individuals and then you release them again, and then you try to capture them again and see how many marked individuals you recaptured. So this is called mark recapture methods. And it's a really cool method because you can actually do some math to determine the total number of these individuals in the entire population based on how many you collect the first time and mark. And then when you release them, they mix with the whole population. And then you go out and you do another collection event and you see how many marked individuals were recaptured. So the math is kind of, it's not too complicated. It's just this, this equation here where N is the total population size. So this is unknown. We're going to try to estimate it. And the way we estimate it is we go out and we collect some animals. And that is M sub 1, the number of animals marked in the first sample. So we go out, we collect a bunch of organisms and we mark them all. Then we release them back into the population. And then in N sub 2, that one over here, is the total number of both marked and unmarked animals collected in the second sample. And then M sub 2 is the total number of just marked animals captured in the second sample. So the fraction of animals that were marked that we recaptured divided by the total numbers of animals in the second capture is equal to the number that we marked in the first sample divided by the total sample size. And so how this works is, for example, say we captured 312, I don't know, rough-skinned newts, and we marked them somehow and we released them into the pond. And then we went back and the second time we captured 328 individuals, but 240 of them had our original marks. Then we would just do some fancy math here. We would put all of these numbers in. So 240 divided by 328 is equal to 312 divided by N. We would cross multiply. So we get N times 240 is equal to 312 times 328. And then we just divide both sides by 240 to solve, and the answer is 426. So what that means is that the total population size in the pond, we would estimate at about 426 individuals. So this is a really cool method for being able to estimate the total population size without having to exhaustively sample every single individual in the pond. Okay, so moving on to reptiles. There are lots of direct observations that you can make. There are also lots of behaviors you can observe. And then there are some indirect methods. So maybe you're looking for tracks or shed skins or dung, or you're using some kind of a camera trap to make pictures. And then there are some capture techniques. You can use some of the same capture techniques for reptiles as you can for amphibians, but some are a little different. These are pictures of Dr. Emily Taylor, who you can follow on Twitter, always posting really cool pictures and things about snakes. So she studies rattlesnakes in particular. And here you can see her using a grabbing tool to kind of interest the snake into going up a plastic tube. And once the snake, the dangerous parts of the snake are in the plastic tube, then you can make really careful observations about the snake and it's not a danger to anyone. This is another tool that you can use in herpetology to capture lizards and snakes. It's kind of like a lasso where you can cinch the lasso down around your organism. And here's another scientist that you can follow on Twitter. This is soon to be Dr. Erin McGee, who is AfroHerper on Twitter and runs a pretty popular game called Find That Lizard. So if you're interested in following herpetologists on Twitter, it's kind of a fun thing to do. Okay, so birds, we have the same kinds of things. We can make direct observations, this time mainly using binoculars, spotting scopes, or telescopes, taking photographs and videos. Often you need some kind of a hide or a blind to stand behind so that you don't scare the bird. So here's just some really cool blinds that exist in parts of the country. And it can be really hard to count birds in flight or large aggregations. So often photographs or videos are taken so that more intense measurements can be made. You can also indirectly make observations about birds, things like nests, droppings. And there's a lot of really great big data out there. There's lots of annual census data, like the Christmas bird count, the breeding bird survey. And then we're going to hear about some of these methods in other ways, but there are timed species counts and point counts and line counts and flush counts. So these are different methods. You have different ways of doing this. You might walk through an area for a specific amount of time. You might walk along to different points and at each point stand for a specific amount of time. You might walk along and flush birds using different methods. These are not commonly used because they're pretty disruptive. And either, any of these methods can involve observing birds or listening for birds. So a lot of bird methods involve songs and sounds, and sometimes sound recordings. Bird capture techniques are often highly restricted by law. And so handling birds, capturing birds is not something you want to just do as a person. But as a scientist, you can get a permit to do these things. To capture birds, you can use what are called mist nets, really fine nets that the birds kind of just get a little bit tangled up in, and then you very carefully extract them from the net without hurting them. And then you can band them, you can weigh them, you can take measurements, determine their sex and things like that. There are these propelled nets that I talk about in the book where the net either like closes or shoots like with a rocket and captures birds. I've never seen these in use before, but they seemed a little interesting. And then we talked a little bit about marking individuals. There's a really complex system for marking birds with these different colored bands. And, as you learn more about it, you learn that there are actually lots of different unique combinations of colors that can help you determine who the individual is, where they came from, and what their sex is. And then moving on to mammals, again, you can make direct observations, although now you need to be a little bit more careful about your own safety. Often, you might use night vision goggles or red lighted flashlights. Camera traps are really useful for organisms that can sense, human observers and will avoid areas. And you can still use mist nets for things like bats. You can also measure lots of tracks or other signs, scat, burrows, runways, latrines, hair. Mammals leave behind lots of clues as to their presence. And here's a pretty interesting kind of like trap. It might be baited. And so it gets, little animals come in and they walk across the ink pad, and then they walk across the paper and they record their little tracks on either side of the ink pad. So it's kind of a fun little trap. Capturing mammals, again, often restricted by law. Again, you can use mist nets for bats, but you can also use things like bat detectors that are sensing the sounds that bats make as they ping for their echolocation. And then you can capture some small mammals live in things like Longworth traps or Sherman traps that are, again, they're baited. You want to put these out in the cool of the evening, and you want to get out there really early in the morning to catch, check the traps before they get hot. And you want to be really careful they don't get too cold. So the smaller the organism, the more potentially deadly sitting for a long time in a trap is. You want to be really careful about trapping mammals. And again, you can mark mammals, and you can capture, mark, and recapture, and you can determine population sizes. You can also put out hair snares, and you can do work like DNA work to estimate, population genetics in a population. Here you can see Rebecca Paradis from the Lower Elwha Clallam tribe using a telemetry device to monitor. She actually has a whole bunch of, oh shoot, they're fish. They're not the kind of fish you would normally think of. I think they're freshwater lamprey. Sorry, I was thinking eels, but that didn't sound right. They're freshwater lamprey that she has tagged along the Lower Elwha River, and she's using a telemetry device to determine whether any of her lamprey are nearby. And this is from a field trip a couple years ago. So, but you can also use telemetry for lots of people, you know, study elk movements and cougar movements using telemetry and radio collars. All right, so those are the methods for some mobile animals, and we'll continue this in part 3.